The microbiome is our invisible super organ. Yes, it's true. Why? Because this is a super organ dependent on any type of the organ, dependent of which type of function it's doing. But all of us, we already aware that microbiota play in our body very crucial role. Let's start from something. To start to describe the function of microbiome, we need to rather to be focused on a definition and terminology, because different people differently understand terminology, because it's quite new field, and we still used, a lot of us, we still used microflora, but microflora is probably not correct. It's actually outdated term, and we need to turn now to microbiome and microbiota. Will be better to use these terms, but we need to understand what means microbiota and what means microbiome. When we are telling microbiota, we mention, we are clarifying the microorganism which we can cultivate. So it's cultivated microorganisms. Compare to the microbiome, and the microbiome, it's some sort of set of genes, and which we can isolate from all the microorganisms, cultivated, non-cultivated, of precise particular niche. It can be argued then microbiota or microbiome is invisible super organ, because it's, let's say, located in specific niches, and it's also carry out some specific function. Okay, when we speak about microbiome function, we need to understand that they very rare, so they are very, very different, and they are really comprehensive. When we are speaking about microbiome, what it's doing for our host, so we can speak weeks, but be very precise, we need to mention regulatory function, because they really regulate gut composition. So they are carry normalization, they support immunity, especially local immunity. And this is local immunity is very important in order to prevent our host against of chronic inflammation. For example, chronic inflammation can be a trigger of different type of disease, and usually it's non-infectious disease. But we also need to understand that any type of infectious disease can be caused when we have a dysregulation or a dysbiosis of microbiome. We need to remember that around 75% of a structure of immune structure and immune cells are concentrated or located in intestine. And they are closely connected tissues like gut tissue, or if it's not intestine, it can be nasal associated lymphoid tissue, which actively interact with microorganisms which are promoting so-called immune tolerance. So it's local tolerance, or I would say immune homeostasis, which can prevent different type of inflammation, including chronic inflammation, instead of healthy physiological inflammation. And actually for today it's proven that if composition of microbial cells are changed, so and we have some stage like let's say dysbiosis, so it can cause sooner or later, it can lead to let's say to the some damaging of intestinal epithelial cells, and this is epithelial cells can can cause increased permeability. And I guess anyone already aware about specific term leaky gut. Leaky gut means we have a problem because we have a different permeability for a different microbes, and it's not really very good, and it's actually the trigger for a variety of non-communicable diseases, also IBD, immune bowel disease. So the main function then will be not only regulatory or immunomodulation function, but it will be also function to maintain balance of immune system. And the main role of our microbiome superorgan will be to prevent different type of infection disease, because It's support some sort of self-tolerance and not response to different factors which can affect the microbiome. So when we are speaking about a microbiome, we need to mention this the different composition of bacteria. They are responsible for a certain composition of bacteria. They are responsible for the absorption of a nutrient, different nutrients. So absorption itself depended which microbes we have there on epithelial surfaces, because efficacy and efficiency of assimilation determined by different bacterial species, but not the separate species, but their composition. And of course, each species will produce different metabolites products, including vitamins, amino acids. But uniqueness of intestinal gut microbiome actually in ability to ferment some indigestible fibers, for example. So we cannot eat that fibers, but microbes doing them, available for us. So there are a lot of nutrients and nutrition components and biological active components which microorganisms are producing. Among of them is well-known short-chain fatty acids, including acetate, propionate, and butyrate. So we know that mainly acetate, they plays an important role in the regulation of metabolites and extraintestinal tissue, including cholesterol. This is very important for people, also for animals, to keep their weight, proper weight. Propionate important for glucose homeostasis, and it's regulate glucogenesis, and also it's participate in satiety signaling. And butyrate, it's capable of activating intestinal glucogenesis as well. And the last one is very important, not only for metabolic balance, but also for energetic balance. So, okay, so can we change microbiota very easily? Can we keep it healthy? Is it very easy to destroy it? Actually, no, because the microbiota of mucosal sites relatively stable. Still, why? Because they can form a biofilm, so they are originated from a host. They have a very close connection with tissue, with epithelial cells, and they are really resistant to various influences. So they are keep us healthy, but still we need to understand that microbiota is not something not changed in a composition, because there are a lot of external internal factors are influencing on microbiota. And also we need to understand that microbiota is changing itself during our age, growing, style of life, physical activity, our mood. So we understand that different factors like the main affecting factors will be nutrition, food quality, stress, style of life. Everything will be affecting our microbiota. But the most probably dangerous for microbiota or microbiome, there are external influences like toxic substances, pollutants, antibiotics. Okay, can we achieve to reconstruction or a recovery of microbiome? Yes, it can be done, but here we need to really keep in mind that first of all the different type of microbiota is strongly individual. So it's not necessary it's not a goal to repair it, to restore the microbiome to original stage. So we need to understand there are a lot of changes, but we need to keep it healthy, in healthy composition. And when we mention healthy composition, we need to understand that it's not each one species we need to keep separately, because they are composed together. So we need to be oriented on that composition, but keeping in mind that we need to be oriented in their function, because always the function makes sense. So, and the norm is strictly individual. So when we want to keep our microbiome healthy, we need to remember our individual norm, we need to remember that it's not different strains, but it's composition of them, and we need to keep it in a relevant condition to support a self-healing microbiome process. Okay, still, changes in nutrition will be most crucial. What can affect our microbiome, because we are not feeding ourselves, we are feeding our microbes. So that's why it's very important to provide a very proper nutrition. At the same time, to understand which nutrition, which bacteria, which microbes, which biopreparation we might use for recovery of microbiome, we need to understand that better to use IT instruments today, because only IT instruments or algorithm can help us to select individual diet or individual nutrition or individual probiotics which we can use. Does this means that probiotics, for example, are not very important for usage? Not, of course, because probiotic is the supplement actually to our food. Actually, there are a lot of food, like fermented food, we can treat as postbiotics. So this is no any competition. They can be together. The goal is together with the food and the probiotics to treat our host. So their role, probiotics, become nowadays probably more important, because we based on microbiome study, we understand now much more better what exactly doing probiotics, prebiotics, symbiotics, and their role and their function can be predictable. And everything what we would like to say, that all this preparation, nutrition, factors need to be calculated on a principle of evidence-based, not only in medicine, but also in veterinary practice. So today, it's not actually the problem, because today we have a variety of different preparation. Variety is really amazing. We have a lot of different strains, we have a lot of different composition, different bacteria, but still, wherever they are so different, we have a microbiome study, we have a biopreparation of next generation. In majority of them, we still can use the old classic classification like probiotics, prebiotics, symbiotics. When we understand that probiotics are live bacteria, prebiotics is something what will be feeding rather microorganism, and symbiotic, some preparation which are combining or using a prebiotic component and probiotic component as well. So when we prepare new probiotic, prebiotic, symbiotic, we need to keep in mind that they need to be synergetically acting. So and also we need to understand which type of bacteria from which source we need to isolate. So it cannot be randomly selected species, because we know that lactobacilli or bifidobacteria is very good. We need to keep in mind what we would like to do with this bacteria, from which niche they isolated. Actually, for what exactly we are going to use them, and which goal we are going to, which problem we are going to solve. So the main issue, they need to be able to restore the balance, and as we mentioned, the balance is very specific. So to apply or symbiotic on next generation probiotic, we need to keep in mind what exactly we would like to get. So which type of function we would like to repair. So first of all, probably it will be the function, it will be about probably immune regulation, it will be self-balance, supporting, maintaining of other commensal bacteria, it will be inhibition of different opportunistic bacterial pathogens, and the symbiotics actually probably it's most important, and even better can be applied to solve this different complex action. And it's extremely important that these bacteria, which can, bacteria, but not only bacteria, but also prebiotical component, cannot destroy themselves. So they need to be very harmonically composed, and they need to synergetically act. So it should be some sort of internal balance, and they then can provide or can be recommended for the prevention any type of the disorders which is connected with the so-called dysbiosis. So even there are a lot of some infectional processes now connected with the disbalanced microbiome. So the modulation of the local immune response, which we call like self protection, or physiological information, can help the host to prevent a different type of disease, not only non-infectious disease but also infectious diseases.